and people eat things day in and day out. Diet is also a behavior. It's culture, economics, planet health, availability of the foods, and lots of other reasons that people choose certain foods. Many of you are probably familiar with this very well-known figure that was first described by Hanahan and Weinberg about 25 years ago. And they describe the hallmarks of cancer through these mechanisms that you can see on the slide, showing that carcinogenesis is a multi-step process that is not necessarily linear. And importantly, many of these hallmarks might not be equally weighted across cancers. Some cancers might have more influence from proliferative signaling, and others might have more influence from evading immune destruction. Across all of these, there are nutritional inputs. Very recently, the hallmarks were updated. In the 2026 Cell publication, Hanahan wrote, while broadly embraced by the cancer research community for its simplicity and utility in offering common principles underlying the daunting complexity of this disease, other frontiers of cancer research were suggested and added additional parameters. So with that, this new model that just came out within the last month or 6 weeks does recognize these additional inputs and potential modulators of the hallmarks. This includes nutrition, body habitus, obesity, and the microbiome. So in essence, we have a really complex exposure, which is nutrition, and we have a really complex disease, which is cancer. And in the cartoon here, you see this person trying to explain a very simplistic A to B model. And then the person at the door saying, yes, not quite like that. And if you look at the outside, there's multiple twists and turns to a system. And the outside is often how I think about nutrition and cancer. A lot of twists and turns. It isn't necessarily A arrow to B, exposure to disease. It's really complicated. So in this system, can we possibly make any progress? One way to think about diet and cancer is an image with puzzle pieces. Can we think of nutritional exposures as pieces of the puzzle, and can we put together just a few pieces of the puzzle? We might not get the whole puzzle, but if we can get a few pieces, maybe if we're lucky, we'll get a corner piece, maybe we can make some progress. Because it's so complicated, I like to use multiple approaches to studying diet and cancer. I do work with observational data, experimental nutrition, and nutritional biomarkers. Each has their own pieces of science that contribute to making the puzzle pieces. So I'd like to give an example for how I tried to integrate observational and experimental data to address a research question related to nutrition and cancer. And that question is red and processed meat consumption healthful, harmful, or neutral for cancer prevention? In 2015, the International Agency for Research on Cancer, or IARC, declared that red meat is a probable carcinogen, a group 2A carcinogen, and processed meat is a carcinogen, a group 1 carcinogen. So you don't get declared a group 1 without pretty consistent and strong evidence and solid biology. Well, what is the biology? In that 2015 IARC report, these are some of the possible reasons that were put forth as to why red meat and processed meat might be carcinogenic. And remember that some of these compounds arise during food preservation and preparation. So there could be formation of specific compounds that may damage cells during DNA. There's a compound called FIP that many of you have heard of, and it's thought that FIP might induce DNA damage. There are nitrosamines, advanced glycation end products, heterocyclic and polyaromatic hydrocarbons. There may also be inherent properties of the meat itself, including heme iron, saturated fat, TMAO, which is produced by the gut bacteria. These may also influence cancer risk when red and processed meat is consumed. Despite some of this biological evidence, the links between red and processed meat remain quite controversial. So back in 2019, there was a series of systematic reviews and meta-analyses on red and processed meat and multiple outcomes. And the full series was published in the Annals of Internal Medicine. I took screenshots of just 3 of the papers, but there was about 6 or 8 of these papers. The series of reviews was very controversial because the conclusions drawn in most of these was different than much of the current thinking and evidence at the time. And most of these systematic reviews concluded in their concluding statements that red and processed meat was not harmful for cancer, for cardiovascular disease, and even the one called health-related values, they said, oh, well, people really like eating red and processed meat. So it's part of their value system. They shouldn't have to give it up. And then many of you may know that the recent 2025 dietary guidelines for Americans omitted any mention of consuming red and processed meat at all, completely omitted. So can experimental nutrition in humans help us understand the biology? And a good research design that many of you are involved in is controlled feeding trials. These are some shots of our human nutrition lab at the Fred Hutch, and I was able to see your facility today. So it's really wonderful that so many places are doing these types of studies because they can really help us get at the mechanisms. So here's the study design that we set out to do. We call it the Eat Well study, the effect of red and processed meat on the metabolome and the microbiome. Diet A has no red and processed meat, and Diet B is identical, but does have red and processed meat, and it was about 1 to 2 servings a day. We thought that we did not want to completely overload the experimental diets in red and processed meat. We wanted to offer diets that were very consistent with what most people in the US were consuming. So we patterned the number of servings that they would get in this controlled diet off of NHANES data and what most people were eating. So we had 20 participants in a crossover design. And as many of you know who do these kinds of studies, when you do a crossover design, it effectively doubles the sample size because everyone acts as their own control. And you can see all of the specimens that we had them collect along the way. And here are some data collection details that we had them do. I always like to give a big shout out to participants because, as you all know, we can't do our studies without them. And sometimes we ask them to do a lot. And so we're very appreciative of all of their contributions and their willingness to be patient with us. They picked up their food every few days. Any uneaten food was returned and weighed. They also had daily checkoff forms that they brought back with the coolers with their uneaten foods. And from the food weighbacks showed that we had really excellent compliance. About 92% of all study foods were consumed. I want to give you an example of one day's meals on each arm. We did have a 7-day rotating menu, but this is just an example of Diet A and Diet B. And you can see that they were essentially exactly alike, except one arm, Diet A, had no red and processed meat. Diet B did have red and processed meat. And making these types of foods made it a pretty easy swap out. So making a turkey and veggie chili, it was pretty easy to swap that out and make a beef and veggie chili. So that allowed us to just really test what the beef was doing. We adjust the calories based on each person's calorie needs. So we have tiers starting at 1,800 calories and it goes up to about 2,800 or 3,000 calories a day, depending on the person's calorie needs. Today, I'm only going to talk about the metabolomics because I have a graduate student finishing up results on the microbiome, and she should be the first one to get to present those someplace. So she is working away on that. We've collaborated with the Northwest Metabolomics Research Center at the University of Washington for a number of studies. They're a longtime partner in our research, and they have established protocols for data processing, metabolite identification, and creation of targeted panels. We collected both serum and urine for this study, and we used a targeted panel for everything except for the GCMS urine, and that was untargeted. So here are some of the selected results for the plasma, and I'll walk you through this, how we did this. So the left-hand side on this slide, these are some of the aqueous and the lipid results. And then the middle shows the ratio of diet B to diet A. So if the ratio is higher than 1, that means that that metabolite was higher on diet B. So we did come up with a number of metabolites that at the end of the 2 feeding periods when we did the analysis, these metabolites were higher after consuming red and processed meat with everything else in the diet being exactly alike. I didn't list all the phosphatidylcholines and the phosphatidylethynolamines because there was 15, but their ratios were in this range, approximately. We did adjust for diet arm, feeding period, sequence, and the baseline value. And then we did do multiple comparisons because we have so many things you're testing. You have to do some multiple comparisons for these types of studies. And then for urine, these are the linear mixed models, also adjusted for the same things. There were only a few metabolites, to our surprise, based on some other work we're doing, that showed up for urine, and none of them survived the multiple testing approach. So that just surprised us a little bit because we expected more to survive from the urine. So what do these results tell us? Can we link any of these feeding study data to carcinogenesis? Because of course, in a short-term feeding study, you don't have any cancer outcomes. We need studies that have long-term follow-up of participants to be able to link to cancer outcomes. So observational data give us information about usual intake over time and relationship to cancer risk, or any chronic disease, but I mostly study cancer. A lot of observational cohorts are thought to be closer to the real world because you're following people over time, so it can be broadly applicable to population prevention. So quite a bit of the observational work I have done comes from the Women's Health Initiative, where we have been studying diet and cancer in a large cohort. So I'll tell you a little bit about the Women's Health Initiative, or WHI, in case you're not familiar with it. Our slogan is changing the future of women's health. For the women, when we recruited them back in the 1990s, one of our recruitment slogans was, "Be part of the answer." And that really resonated with the women. They want to be part of the answer. So for some of you who might be graduate students or newer to science, prior to the Women's Health Initiative, there really was very little scientific research that was focused on women, especially clinical trials. Mostly results from men were extrapolated to women. And there were all these crazy thoughts going around about why women shouldn't be research participants and why they can't be in clinical trials. Just very outdated thinking. And Dr. Bernadine Healy, who was the director of the NIH at the time, she had to fight and fight and fight and fight to get this funded. So we're very grateful to her for doing that. So between 1993 and 98, we recruited 161,808 postmenopausal women who were aged 50 to 79 at the time. We had 40 clinical centers throughout the United States, and at the Fred Hutchinson Seattle, we had one of the clinical centers, Plus, we have always served as the data coordinating center. We included a focused recruitment for women underrepresented in research. So we had about 20% of the participants were Hispanic and Black women. And that was, about 2 or 3 decades before a lot of other studies realized that we need to have studies that are more representative of what the nation looks like. Today, we actually have over 40,000 women who are still alive and participating. They participate in ancillary studies, they answer annual questionnaires about their health, and they come to our participant webinars. About 13,000 of them are over the age of 90, and 600 of them are over the age of 100. So they are truly amazing women. Here's a little bit about the design. The Women's Health Initiative was designed as 3 overlapping clinical trials: a hormone therapy trial, a calcium vitamin D supplement trial, and a dietary modification trial. Women could be in 1, 2, or all 3 trials, and you can see the numbers here. So in the dietary modification trial, over 48,000 women in this trial, across the country, really quite remarkable. And women who did not want to be in one of the trials, or if they were not eligible because there was more strict eligibility criteria for the trials, they could opt to be in the observational study, and there was nearly 94,000 of them. So a little bit about the timeline. In 2005, when all of the trials ended, then everyone converted over to being in an observational study, and that still continues today. So dietary assessment has always been a big component of WHI, not only because of the dietary modification trial, but also the observational study. And we did quite a bit of dietary assessment. Some of the other cohorts have only assessed like a one-time baseline dietary assessment, and we did it periodically, and we've done some more in later years as well. So women who were in the observational study, they completed a food frequency questionnaire that was designed specifically for WHI. It had many cultural and regional foods on it because we were at 40 centers throughout the US. And then those who were in the dietary modification trial, they completed it at baseline, year 1, and then every 3 years. So every year there was a rotating 1/3 who completed it until the trials end. So we have authored, literally hundreds of WHI publications on diet and cancer. But one of the things that we have thought about, and I've done quite a bit of work on in my career, is this problem called measurement error. And measurement error is the difference between the true value and the measured value. And you can see that illustrated here at the graph on the right. Random measurement error is like noise. It just is noise that you can't really get rid of, but sometimes it can be remedied with repeated measures, and then you can take a mean, of all of those measures. For example, multiple 24-hour recalls, there's going to be noise from recall to recall, but if you average them out, that gets rid of some of the noise. Systematic measurement error is very different, and that type of measurement error distorts the associations, and it cannot be remedied with repeated measurement. And systematic measurement error is a concern in dietary assessment, and for some of the studies that only rely on self-report, that may be responsible for some of these differences that we see across studies. So in the Women's Health Initiative, we've been trying to use biomarkers to help correct for some of this systematic measurement error. And we first started thinking about this towards the end of the dietary modification trial. We knew it was going to end in 2005. So in 2004, we started thinking, what are we going to do? Because at that time was when the evidence was just emerging that dietary self-report had both random and systematic measurement error. And we needed methods to be able to properly interpret the dietary modification trial results, which we were going to have about a year after that time. Now, recovery biomarkers are probably the gold standard, the best way to have a nutritional biomarker, because in those biomarkers, intake equals output. Unfortunately, we don't have very many of those, but that was where we started. And so we used some recovery biomarkers as an approach to help us start to understand this phenomena of misreporting. So we started out while the dietary modification trial was still going on, and we called it NBS, or the Nutritional Biomarker Study. And we enrolled 544 women across 12 of the centers across the US. They were still participants in the trial. About half of those we enrolled were still in the active intervention, half were in the comparator arm. Basically, what we did is we ran them through, excuse me, a W-labeled water protocol to get a measure of total energy expenditure, which in weight-stable people, that's approximately equivalent to energy intake. We collected 24-hour urine so we could get a measure of nitrogen, and then you apply a fraction, you get protein intake, and so on. So we did that. And then a few years later, we got funding to do the same thing in the observational study. And then a few years later, in Seattle only, we ran women through this whole protocol while they were also on a controlled feeding trial. And in that controlled feeding trial, it was different than like testing a diet. We wanted to test what biomarkers appeared when they were on their usual diet. So before they started the 2-week feeding study, our study dietitians collected 4-day food records from them and did a lot of documentation on brand, size, all these other things, cooking methods. And then we basically duplicated each person's 4-day food record as their individualized diet. So it was very expensive, very time consuming, but I think it was a good design. And you can see here at the bottom, all of the biomarkers that we have measured in, over 1,000 of these women. So just to give you a flavor of some of these early findings, which we called discovery, this was in our first study called the Nutritional Biomarker Study. We had all of the women, the 544 women, complete a new food frequency questionnaire when they came in to start this W-labeled water protocol. So they completed many of these over the course of the trial. They were going to complete another one as part of this protocol. So what you're seeing here is their self-reported diet, the energy from the food frequency questionnaire. And you can see for the women on the intervention arm of the dietary modification trial, they report on average about 1,145. Did this go off or is it still on? Okay. Calories per day. And the comparator arm, a little bit more. Does this mean they're underreporting in the intervention arm? Maybe they weren't reporting everything since they were supposed to be on a low-fat, high-fruit and vegetable, high-grain diet. We don't know. But what we do know is when we compared it to, oops, sorry about that. When we compared it to the recovery biomarkers, the energy from the W-labeled water, we see a really big difference. So, 500 more calories difference. Same with the protein. There was also underreporting, but not quite as dramatic with the protein as with the energy. So we did a lot of work, and long story short is that we showed that there was systematic reporting, and it was related to participant characteristics. So there was significant underreporting for women who were in the dietary modification arm by BMI. Women who had a higher BMI underreported more than women who had a lower BMI, and Black and Hispanic women. And for the Black and Hispanic women, we think that maybe we didn't do a good enough job creating that FFQ in the first place, that maybe we didn't have enough of the foods that they normally eat. And to show you how it's not always underreporting, but it's distortion, we found statistically significant over-reporting for age. So women who were older tended to over-report, not under-report. And then also for other race ethnicity, which in this case was Asian, Pacific Islanders, and Alaska Natives, and then also current smoking. People who were current smokers also tended to over-report. So now what do we do? Okay, we know this is happening. We have some biomarkers. Now what do we do to try to apply it to these cohorts that have all these, FFQs? Well, this is why I like team science, because I never could have thought of this myself, but I work with some really brilliant biostatisticians. And so they came up with this method of regression calibration, where if you measure the biomarkers in a subset, as long as that subset is representative of the entire cohort, then you can come up with an estimate for the self-report that considers all of these variables that we knew were affected by the misreporting. So we did all these simulations to come up with these calibrated equations. And then what actually is used in the self-report when we're doing diet disease association models is not what they filled out on their FFQ, but it's a predicted value. It's a predicted value that considers the doubly-labeled water in the subset and all the participant characteristics that are associated with the misreporting. So that's why team science is great, because I never would have thought of this. And they never would have thought of some of the nutrition things that those of us in nutrition would have thought of. And so this is just an example of how it works. You see over in the left-hand column, BMI, the three categories that we're all familiar with. And then self-reported energy from FFQ. So they're all about the same, a little over 1,400 calories a day. And we have to think to ourselves, do we really think that's true across a big range of BMI? Would they all be consuming a little over 1,400 calories a day? Well, maybe not. Maybe something's going on there. So then we see the next column, what the biomarker is telling us. And the biomarker, again, is telling us that the self-report was underreported because the so-called truth is a lot higher. So then through this regression calibration method, then we come up with these predicted values for energy that seem a lot more realistic, given the BMI. So that's just an example of how it has worked. So if we go back to the design for these biomarker studies, I want to talk a little bit more about the feeding study. So remember, I mentioned that everyone had their own personalized diet from their food record, and we fed that. And then what I'm going to talk about with regards to the red and processed meat is some of our metabolomics data. So we've done a number of these analyses with lots of different dietary factors. We try to use the feeding study of sort of discovery with the metabolomics. And then we use the observational data as to develop the calibration equations. And then we try to apply it to the whole WHI cohort. So how can we use these data and this technique to try to understand the association of red and processed meat consumption in older women with chronic disease risk? And so here, these are the metabolites that we found that were associated with red and processed meat from the feeding study. And when we make these models, like sometimes you'll read a paper of nutritional biomarkers, and there'll just be a correlation of the biomarker, with the mean intake of whatever the nutrient is. And if that correlation is 0.4 0.4, 0.5, then everybody calls it a day and says, oh, well, then the self-report must be pretty good because we had a good correlation with the biomarker. What we try to do is we try to get a model R squared of at least 36 for the biomarkers. This is the explanatory variation in the intake because we know what the intake was in the feeding study. And we try to have it at least 36% or above, which is a correlation of 0.7, because that's what we get with W-labeled water. We want to set a really high bar. So if W-labeled water has an R squared of 0.36, correlation of 0.7, we want to set that bar high. And so here, with these analyses, and we use a cross-validated method where we leave some out and do a cross-validation. And our cross-validated R squared here shows it's explained 45% of the variation in the intake in the feeding study. So we were pretty pleased with that. Now, before I go on to show some of the cancer results, what I've shown in red here is the biomarkers that emerged in the previous feeding study that I described, where we expressly fed bread and processed meat. So it was nice to have some, overlap. It wasn't the same lab, which is also good, because if I'd sent it to a different lab at UC Davis or someplace else, it probably would have been completely different answers, because each metabolite panel is different across labs. But it was nice to see some confirmation of these biomarkers in our WHI study, because again, it was based on their usual intake. So it might not have been 1 to 2 servings a day the way we had it in the other controlled feeding study. So then back to the same approach, we're going to use those biomarkers that we discovered and then calibrate the self-report. And what did we find? This is what we found for cancer outcomes in the WHI. And I've shown, without the biomarker calibration and with the biomarker calibration. And the models are the risk above the median intake of 42 grams per day. So it's mostly null if you just use the FFQ with no calibration. And it's a little bit higher. We're never going to get a relative risk of 2 or even 1.5. But even to having these relative risks that are not null, we thought that was pretty good and confirms that red and processed meat intake is likely associated with risk of these cancers. And we had plenty of cancer cases because we've been following these women for over 30 years. All of our outcomes in WHI are adjudicated with medical records, so it's not based on self-report. We get the medical records to confirm it, and then we adjust for a number of variables that we have been collecting over time. So now I want to move to another topic. So I hope you don't mind switching gears with me for just a moment, and that is obesity and cancer. This is something that I've been working on for several years, and you heard, some from Katie this morning about the TREC centers that I was also involved in. And the question I was interested in answering was, how does eating frequency relate to obesity and cancer? And actually, this rose out of some of my TREC work in my TREC project. Well, you probably already know this, but obesity is actually linked to higher risk of 13 cancers. And for these, obesity is measured by high BMI. You can see all the cancer sites here. There's growing evidence to suggest that obesity may be linked to the rise in early onset breast cancer, especially in Black women, and rising colorectal cancer in young people. Although those are complicated stories, and it's like lots of things at play. But 13 cancers is a lot of cancers. So my question that I was interested in is when to eat. There's lots of different studies out there right now examining meal timing, circadian rhythms, all these different things. For example, when to eat has lots of different definitions and ways to operationalize it. So time-restricted eating, or TRE, is eating within a specific time window each day, usually restricted to limited hours, like 8 to 10 hours a day over 24 hours or something like that. There's also something called intermittent fasting, where there's no food eaten during certain hours of the day or even days of the week. You're probably familiar with the 5-2 intermittent fasting, where you can eat whenever you want, whatever you want on 2 days, but intermittent fasting on the others. I was interested in frequency of eating. So I have defined this as how many times someone eats over a 24-hour period. So in layperson's term, you can think of this as a meals pattern versus a grazing pattern. And I got interested in this when I was thinking about the physiology. And sometimes new scientific ideas can emerge when you least expect it. And in my TREC project, which was a randomized crossover feeding trial in our Seattle TREC Center, study participants had to fill out a baseline food record before they entered into the feeding study, and they had to have a baseline normal, blood glucose to be eligible. And I kept seeing, even in these younger people, we'd look at their food records and they would show sort of this grazing pattern of eating. And most of them were coming in with these, moderately elevated blood glucoses. And these were people who were young, healthy. They had a normal, body weight, but they were like in the mid-90s. And I just started wondering, well, what is going on here? So I came up with this diagram that I've been kind of using over the years since then, that, someone who eats sort of a meals pattern, their blood glucose goes up, and if they're healthy, it goes back down. They eat again, it goes up. They're healthy, it goes back down, and so on. But someone who might be eating all throughout the day, a little bit here, a little bit there, their blood glucose and insulin might stay elevated. So what does that do for health? And so that has led to a series of studies. I keep wondering what's happening with the counter-regulatory hormones. If glucose and insulin are always staying up, if someone's always kind of nibbling here and there, eating every 2 hours, what's that doing? So as part of that study, we did a little pilot where we had 10 people, or we had 20 people, 10 on each arm. This was some TREC developmental funds that Katie remembers. And we had people, randomized eating their own foods, because we didn't have a lot of money, to either a meal pattern or a grazing pattern. And we had them at the end of whatever the time period was, 10 days or whatever, we had them wear a CGM. And what we found out, even in this small study, was that the area under the curve over those 4 days for blood glucose was higher on the grazing period than the meal period. So we kind of took that and we wrote a study called The Frequency of Eating and Satiety Hormones, or the FRESH study. It was a randomized crossover trial, but it wasn't a feeding study. But we tested high versus low eating frequency 6 times a day versus 3 times a day. It was the same food, same energy intake on both trials. The meal plans were designed by our study dietitian, and she designed meals and eating plans at 8-calorie tiers to accommodate the energy needs. They were free living and cooking their own foods. And then we also did end of study postprandial testing. So this is a little bit more of the design of what we had them do. And today, just because I thought I couldn't present everything, I thought I would present the end of study postprandial testing. And Katie is clapping because now she's an assistant professor at Ohio State University. Here's a postdoc with me, Xiaochen Zhang, and she worked with Katie as a master's student. So it's a small world in what we do. So this is what our end of study postprandial testing was. If they were on the high eating frequency arm, they had the same size meal, at breakfast and then at lunch. If they were on the low eating frequency arm, we gave them, the same breakfast but split into 2 meals. So they would eat some of it at 8 o'clock, and then they would get to eat again at 10 or 10:30 and so on. We took blood throughout this time period, and then they also filled out self-reported measures of appetite. So I'm going to start with our ghrelin results, and remember that ghrelin rises before eating and tells the body to eat. And so here we could test where the more frequent eating compared to the less frequent eating influenced ghrelin. And the bigger meal or the meals eating plan is represented by the dashed line, and the smaller meals or the grazing is represented by the solid line. And you can see that ghrelin fell after eating. It also, or the bigger meal, it also fell after the smaller meal, but not as much. It kind of stayed up. It never really went back to baseline. And so there ended up being a significant difference in the area under the curve, for ghrelin was sustained and higher for the whole 6-hour time period. And that aligned with the self-reported appetite. It seemed like people on the smaller but more frequent meals were a little more hungry. So it lined up with the biological measures. And then now for the postprandial PYY, and this is the hormone that says I'm full. And again, here we still see some differences. We can see at the high eating frequency arm, which is the solid line, had a higher PYY rise that remained sustained and did not drop down. And even when it was time for the second eating occasion, they had not dropped back to baseline. Whereas for the smaller but more frequent meal where PYY rose, it went back down. And these, again, were significant differences in the area under the curve. And that seemed to also align with the self-reported standard appetite measures. So what does this have to do with nutrition and cancer? These measures of appetite. Well, appetite is one of the drivers of food intake. And while there is no one-size-fits-all for appetite management, meal frequency could be a useful tool for some people. There is a lot of anecdotal diet books that tell people, oh, eat every 2 hours and you won't overeat. You'll lose weight if you eat every 2 hours. But at least in our study, we show that that is not the case, that biologically, the appetite hormones tell you, I'm still hungry after I had that little thing, and I'm going to stay hungry. But the larger meals, they were more satiated. So managing food intake and body habitus is an important aspect of nutrition and cancer prevention, even in this age of GLP-1 receptor agonist medications. We still have to help people manage their appetite and manage their cravings. And maybe this is one tool some people could use. And we're still doing many additional analyses in the study. Even though Xiao Chen has gone to Ohio State, she is exploring the macronutrient distribution of each person to see if that has influence over the measures. And then there's a grad student working on sleep. So, and I'd like to thank my collaborators, many of whom you see here, as well as funding from the NIH that has allowed us to do these studies. And thank you again for the invitation to be here with you today. And hopefully we can continue to put some puzzle pieces together.
Questions? Start. Farishi, there was an investigator probably 15 years ago that fed people erratically and then fed them with 3 meals a day. And they literally took all the food from 3 meals a day and just divided it up. And the key there was to make sure that after 3 weeks, they'd had the same amount of food. And the erratic feeding caused insulin resistance and elevation in glucose. And so I guess where I'm going with this is Megan McCrory said that multiple, these books that tell you eat a lot of small meals, her argument is that when you tell a patient to eat a lot of small meals, what they hear is a lot of meals. And every opportunity to eat is an opportunity to overeat. And so can you tell me how it is this message of a lot of small meals more effective when you have a dietitian or somebody helping you not overeat when you go to a multiple meal a day pattern?
Well, that is a really good question. I think we would need a more free living situation where we were following people for a long time to address that kind of thing. Although we did, which I didn't show today, I did use the 24-hour recall data from the Women's Health Initiative. We published this in CDN a few years ago. And the 24-hour recall records in NDSR, have a timestamp. And so we looked at the number of meals that women ate over time, and those who ate more frequently, I think it was 4 or more times a day, they had a higher risk of diabetes.
Because that's advice, right, to multiple small meals. That's been given for a long time. Questions? Yeah, Shami.
So I'm not sure if you can tease out this question, but do you think it's the red meat or the processed meat that increases the risk for cancer? Since it was combined in your design.
Yeah, it is combined in the design. And that's because most of the observational data have combined both of them. And I think it might be because in some study samples, the processed meat, especially the grams, because the grams is less, so the exposure might be a little bit less. We chose to combine it simply because we wanted to shed any light on some of the observational studies. Another design could be just to look at red meat.
42 grams per day of red or processed red.
Yeah.
What does the average American eat? Is that a lot? Is that a little?
That's probably a little bit less than the average American because these are older women in that study.
So in general, then the population is not is on the upper side, the cancer-inducing side of that dose.
Yeah.
Other questions?
For the red meat study, there were a few cancers, but were you able to look at other cancers? And so is red meat, mostly what I'm wondering is red meat consumption or processed meat consumption more associated with certain cancers than other cancers?
Yeah, we did look at a number of cancers, and in the paper, we list them all. I didn't want to make a slide that was like so busy that nobody in the back could read it. So I picked out the common cancers, and I picked out the ones that had the strongest signal. In the paper, we list them all. And we list a few that were null.
Any questions on Zoom or others? Yes, Javna.
Thank you. Great talk. I was looking at the metabolomics panel that you presented, and some of those metabolites, like the phosphatidylcholines, the lysophosphatidylcholines, and there were some, I think, the TCA cycle metabolites on there. We see a lot of the same metabolites in our dietary intervention studies, not just the red meat type studies, but almonds or plant-based foods or high carbohydrate foods. So it makes me think if those metabolites may just be representative of any dietary change, so regardless of what the type of food is, those metabolites are just responsive to dietary changes. And if we should be focusing more on maybe the magnitude of change instead. I don't know if you have thoughts on that, because I see that quite often.
Yeah, I think that's a really good question. And also, a lot of these metabolites have very short half-lives. Some have longer half-lives. So if we're just seeing it as a snapshot, what does that mean? I think we're just kind of at the tip of understanding metabolomics in dietary studies. We're just starting to really understand it because we have to put everything together with all the pathways and
Yeah, so
something
relative.
has a really short half-life. What does that mean for the rest of the organism? Or is a short half-life, if it's a short, potent half-life, does that still have a biological effect? I don't know the answer to that question. That's why we still have a lot of research to do. Maybe you have some ideas on that.
Hi, great talk. This is kind of just a funny question, but I want to know about all of those women who are over 90 and in their 100s. Like, what are they eating? Have we done that research? So, and kind of along those same lines, like, are there any foods that come out? I know like red meat has been kind of singled out because of the studies that have shown that relationship. But on the other side, are there certain foods that, is it lycopene? Is it blueberries? Is it fiber? What's kind of like what's the gold star? Yeah.
Yeah,
What should we be
actually
eating?
I'm working with a young investigator at Wake Forest who has recently submitted an R01 to evaluate, diet and dietary patterns and measures of aging. Some of it's with, the aging clocks, and some of it's with, she wants to do epigenetics on some of the stored blood samples. And what I didn't say is that some of these women then have additional blood samples that we took in 2011 to 2013, and then just 2 years ago, on like 5,000 women, we have more bloods that we can interrogate. So we're starting to think about those, and hopefully, I don't know what's going to get funded these days, but hopefully some of those studies on healthy aging. And we've recently, within the last year or so, created a new frailty measure. I know there's lots of frailty measures out there. And I mean the WHI investigators, not me personally, created a new frailty index. And I have a PhD student in nutrition who is examining, that's where her PhD dissertation is going to be on, diet and dietary supplements over time and the frailty index. So she just turned in her exams, her qualifying exams a week ago, but that's where she's headed next. So hopefully in a few years, we'll have some more to present.
Yeah. So any advice on what's your favorite thing to recommend that we should be eating? Just stay away from red meat.
Well, I wouldn't say that red meat is off the table. It's more of, in WHI, we called this once in a while foods and always foods and sometimes foods. Fruits and vegetables and whole grains are always foods. Things like yogurt are always foods. Water is an always beverage. And then there's sometimes foods which might be, ice cream, some chocolate, red meat. If somebody has a hot dog, it's not going to be a single hot dog. It's dietary patterns over time.
Thank you.
I think also what you're saying is reminding me of the wisdom of funding studies for a very long period of time. Some of us do very intense intervention studies that are short period. We're looking for physiology, but if there's not a commitment from the funding bodies to continue to follow these women, then we are going to lose an awful lot of data. Yeah, thank you for doing this work.
Thank you for the talk. So looking at the eating patterns, do you think, humans evolution-wise, we haven't been around that long. Do you think that's based on how, they used to hunt and you would get something and then you would eat and then you have a period of where you wouldn't find something to eat and you had to, I don't know, resort to grains, whatever you had around. But do you think that's, that's based on that, and that nowadays snacks are everywhere, food is everywhere, so you get this grazing behavior that's probably more new to humans? And thinking about the red meat, if we wouldn't graze that much, would maybe red meat not even have that big effect if we only ate it, so many few times a week and not every day?
Yeah, well, for the first question, I feel like I don't have the experience in evolutionary biology to say, this is how humans ate at one point in time, and this is how it changed. But certainly bringing in, evolutionary biologists and anthropologists to research to help address those questions could be, useful and interesting. I know there's a lot of anthropologists that are really into the biology, and they might be useful to bring into this line of research. And then your second question was, could you say it again?
Yeah, I was just thinking if you would engage more in, these old pattern, or when we started evolving, if you would engage in those patterns, and they probably did hunt red meat, I guess it would just go to you just eat less in lesser frequencies, so you wouldn't have that issue. So
Yeah, possibly, yeah.
Any other questions?
I was interested in how you were able to use the regression models to make the observational data that you got more accurate. And I was wondering if you had any concerns that, say, with your dietary intake forms, that you weren't getting accurate results specifically in like Black and Hispanic women, but for a lot of factors, but potentially because those forms didn't represent the foods that they ate or whatnot. Do you feel, or are you concerned that using regression models to make this data more accurate could cause you to overlook things or change the data in a way that doesn't reflect why the dietary intake wasn't accurate in the first place? And then for future studies, how do you approach making these dietary intake forms more systemic, with less systemic flaws?
Yeah, that is a great question. I think we always have to ask ourselves is what we are doing to the data, is how we are analyzing it, what is it doing? What's it doing to our interpretation? Like even with metabolomics, you get back your lab results and there are thousands and thousands and thousands of metabolites, and you have to figure out a way to do filtering. Same with microbiome. You have to filter some data out because you can't possibly use all of it. You have to do data reduction methods. So if you filter stuff out, are you overlooking something? So I think it's the same principle. We always have to ask ourselves those questions. And in terms of looking forward to how we better assess diet, that has been such a hard thing in our field to try to figure out how do we best assess diet, because it's such a complex exposure. And it may differ depending on someone's economic circumstances, their culture, what food is available to them. So trying to get a tool that works for everyone, maybe isn't the best, but there's a whole organization called the International Society for Dietary Assessment Methods, and they've been grappling with these questions, ever since the society started. So I think it's an ongoing question that we need to continue to figure this out.
Right. Well, we'll stop there. Please thank Dr. Neuhouser.